Hello, everyone. Welcome back to Bio 330, the new improved COVID-18 Bio 330. Same as the old Bio 330, only you don't have to sit in person, you have to sit in video. It's a very interesting time, I have to say. But before we get started, I just want to give you a little perspective on this. This is nothing new. Even though it might be new for you and me, though I had a little experience with this with SARS outbreak in 2004. I was going to a cancer conference and they had to cancel the conference because so many of the people going to the conference were afraid of bringing SARS back to their patients who are on chemotherapy and thus immunocompromised. So I had a little taste of that a few years back, but nothing to this extent. But even though it's new to all of us, this intensity of this, it's nothing new in human history. In fact, there have been many times where we've had to quarantine for various reasons. So we're just a part of the larger picture. So try to make the best of it. Don't be distracted. Hopefully these lectures will allow you to focus a little bit. That's my intention. I don't want to bore you to death. I want to give you a safe space where you can just focus on one thing and keep trying to do that. Focus is a useful tool in your career path. Trust me on that. Before we get started, though, I want to also have you reflect a little bit on muscles. Certainly, muscles are the movers and shakers in our bodies. They contract, they relax, they allow for motion and movement through different compartments of the body. But they're also very, very important in that larger physiologic system that we don't talk about, which is what I would call complex physiologic responses. Things that we do in order to achieve physiologic and reproductive goals that go above and beyond maybe biochemistry. Here we have on the right, we have Jason Lezak, who completed one of the great Olympic performances of all time in the 4x100 meter freestyle. He came in a couple of seconds behind the world record holder in the 100 freestyle in his leg of the relay, and was able to out-touch him at the very end in an extraordinary performance. It's really brilliant swimming. On the other hand, we have another great athlete down here. This is Jessie Diggins, a member of the two-man cross-country skiing team that won the first ever gold medal for the women. Again, an absolutely brilliant come from behind at the very end performance. So muscles serve very, very important roles from a mechanistic standpoint, and we're going to break those mechanisms out, but they also serve a very, very important complex physiologic as well as interaction. One's physique is very much a signature in society. So muscles cannot be understated in terms of their multiple roles in physiology and reproduction. I want to bring your attention just for a second to the now tentative calendar. We're going to be winging this a little bit. What I have done with the new calendar is that I have gotten rid of some of the extra material that I thought would be fun, but because we're in a limited environment, we don't have as much interaction. I'm trying to keep it simple for you guys as possible. What I mean by simple in terms of getting content to you. We're still working on some of the details of how to do assessments. Certainly we have mastering. We're going to be doing some online lab stuff. But we're very much still in the building process of that. So I'm trying to keep it as simple as possible. This calendar is on ACE for you. You can notice that we have some due dates and some things going on. These study groups, these studying and mentoring groups, we're going to have to figure that out too. And maybe what I do is create some kind of a Zoom or some kind of a Skype or something along those lines that we can have questions or do something along those lines. I don't have the technology worked out yet. Maybe what we do is have you submit questions and then I can respond to them, something like that. I'm going to try to figure that out. But I need your help working with me on this. Please pay attention to the calendar if I announce changes or something. please take a moment or two to double check what's going on. So this week we're going to go through muscles, and I have it as 3 lectures. I may try to keep them shorter. I may try to limit them to 30 minutes, and so that may mean more of them, but just in shorter segments, if you know what I mean. So for chapter 12, here are the learning outcomes. We're going to talk about 3 muscle types. We're going to talk about skeletal, cardiac, and smooth, with an emphasis primarily on skeletal and a little bit of smooth at the very, very end in part 3. Cardiac, we'll just talk about a little bit, and we're going to cover most of that in chapter 14. You want to understand the basic cellular and subcellular anatomy of the skeletal muscle. For those of you who are going to be working with muscles, one of the major issues is muscle weakness or muscle inability of muscle function. So we have to be able to work at this level because accurate diagnosis leads to specific interventions. Just like with the nerves, just like with the central nervous system, now we're bringing the effector of those pathways into play in the muscle. Organization of the sarcomere is important in that. That includes the bands, the lines, and the zones, where you find different proteins and the roles of those proteins. What do those proteins do in a sort of haiku level function? The summary map of muscle contraction. Again, double check that page for me in case that's slightly incorrect. Troponin, tropomyosin. We have what we call the big 3 questions. That is ATP status, the configuration of myosin. Myosin is the one that flexes and extends, and the ability of myosin to bind to the other filament known as actin. All of these 3 questions are involved in what's called the power stroke. The troponin and tropomyosin, how we initiate the power stroke, and so forth. And then we're going to go into excitation, contraction, coupling. Muscles do undergo action potentials. And how do we link that action potential, the excitation, to the act of muscle shortening that we call contraction? So some detailed mechanisms. Again, we have to understand them if we're going to understand what causes inability of muscle function. Also in this chapter, and it's a big chapter, we're going to talk about muscle energy supply, particularly when it pertains to fatigue. The role of ATP, we have 3 major roles for ATP. It's not just an energy source anymore. It's serving major functions in the muscle that are all very, very important. Again, as I mentioned a second ago, we're going to look at energy supplies, particularly in relationship to fatigue and what we call fatigue theories. There are lots of reasons a muscle will fatigue due to different molecules, different presence of ions in the skeletal muscle, and so we're going to go over those a little bit. We will get into some classification of muscle, fast twitch, slow twitch, oxidative, glycolytic type situations. This one's a big one right here, this length-tension relationship. This is a big one. One of the most important graphs of the whole semester, so I'm going to talk about that a little bit. Talk about tetanus, what we call complete versus incomplete tetanus. Again, playing back into the idea of weakness and fatigue. Motor units, that's basically a definition, asynchronous recruitment, and then just a little bit about the idea of levers and fulcrums and that sort of thing, isotonic versus isometric type situations. Finally, wrapping up this chapter, we're going to take the skeletal muscle model, and that's our base model for all of muscular contraction, and then we're going to show you the slight differences that we see in smooth muscle. Smooth muscle has a very, very different contraction pattern. Smooth muscle has a much greater variety in terms of the way they function, and we're going to break out the excitation and contraction mechanisms in smooth muscle as a difference from the skeletal muscle. Again, a lot going on in this chapter. This is a big chapter, so we're going to spend a fair amount of time covering all of these issues. So about this chapter, how we're going to progress through topics, we're going to spend the vast majority of our time on skeletal muscle. It is our base model system. That is to say, we're going to compare the other systems to skeletal muscle. Certainly in terms of mechanics of body movement, generating motion, generating force, we're going to look at all three types of major muscle, but we're going to use skeletal muscle as our base, our frame of reference, if you will. Smooth muscle we're going to cover at the very, very end of the chapter. Well, again, using it as a comparison versus skeletal muscle. Cardiac muscle will break out a little bit in this chapter, but mostly we're going to save that for chapter 14. So cardiac muscle gets put by the wayside a little bit, except here in this very, very beginning. So we're going to cover some tissue and cellular anatomy because you have to know these components in order to understand how the contraction and excitation and fatigue processes work. We will get into a little bit of pathophysiology. We'll talk about muscular dystrophy and a couple of other types of basic muscle pathologies to help you, again, understand why we need to understand these pieces in order to understand the interventions that we're going to use to treat those conditions. Certainly the mechanism of contraction is huge, and there are similarities across all three muscle types, but there are some differences as well. So again, we're going to use skeletal muscle as our frame of reference, and then look at smooth and cardiac muscle in terms of the comparison to skeletal. Initiation of contraction, the action potential, what's going on in what we call the motor end plate. We've already seen it though, the story of acetylcholine. If you remember in chapter 11, we have one synapse in the neuromuscular circuit, and that is a nicotinic cholinergic synapse. So we have acetylcholine, and we have acetylcholine nicotinic receptors on the surface of the muscle. So we're going to see some stuff we've already seen in previous chapters. Some theories on fatigue. We have 2 or 3 that we're going to cover. There are more than that, but we're going to cover some of the major ones there. And then a little bit of mechanics. But in this section today, we're going to cover these 2 things, the tissue and cellular anatomy and the mechanism of contraction. What makes skeletal muscle interesting not only from the mechanistic standpoint, but also from a volume and mass standpoint. 42% of the average adult male, depending on how much body fat they have, so the body fat is rising, so that number is actually falling a little bit. But nevertheless, it's a major organ system. 36% of the average female. That means that muscle is a dominant metabolic sink, if you will, metabolic sink for glucose and oxygen and nutrients, but also a major generator of waste products and metabolites that can have extraordinary impact on the human body. As someone who likes to be in a climbing harness from time to time, there is a very well understood phenomenon called harness hang syndrome. That if you hang motionless in a harness for even just a few minutes, we're talking about 10 minutes, 15 minutes motionless, like as if you were unconscious or something, the harness can act as a mild tourniquet. And what will happen is waste products from muscle, like in your large muscles in your legs, will build up, and if you release those products suddenly, as if you woke them up and got them moving after they've been hanging in a harness, people have actually died from harness hang syndrome, from being motionless in their harnesses. It's a very well understood phenomenon. So it's hard to underestimate the importance of skeletal muscle in the pH and metabolic standpoint of the human body. Skeletal muscle does have a major importance, and this is something to note when it comes to differences between cardiac and smooth muscle versus skeletal muscle, is that skeletal muscle can be influenced by hormones in terms of strength of contraction, but it cannot be not directly. That is to say, you don't have hormones initiating skeletal muscle contractions, but you do have hormones initiating smooth muscle contractions in some cases, and even cardiac muscle in some cases. So there's a much more prevalence. There's much more prevalence of hormones in the influence of smooth and cardiac muscle than in skeletal muscle. This indirect influence is the basis for the Marvel comic, The Incredible Hulk. There are stories about the influence of adrenaline in particular on muscle contraction. I even have a story from my science teacher in 7th grade where a tractor fell on him and his brother moved the tractor off of his leg. It broke his leg, but the brother was able to lift the tractor off of him. And when after my science teacher was retrieved, a bunch of guys went out to retrieve the tractor and none of them could move it altogether. So there are indirect stories of influence, but they don't cause direct contraction, unlike in cardiac and smooth muscle. A little bit about cardiac and skeletal muscle. One of the things one of the major technological barriers to appropriate muscle contraction, and you guys have all experienced this in the form of a cramp, okay, very, very painful. What's going on often in a cramp is you're getting asymmetric contraction within the muscle. That is to say, some of the muscle fibers are firing and some are not. And that can lead to divergence from the center line of the muscle. Anytime you're contracting asymmetrically, part of the reason why it's so painful is that it's pulling off center of the muscle. And normally I show you this in class. And so all muscles have to solve a technological issue, which is they need to contract as a single unit most of the time. It's very, very rare that they don't. Either it's pathology or some sort of specialized situation. So if muscles have to behave as a single unit, how do we solve that issue? Well, in the case of skeletal muscle, you fuse the muscle fibers, muscle cells together to form this large Hindenburg-sized fiber. In the case of cardiac muscle, the cells have solved this another way, and that is through intercalated discs. So cardiac muscle is similar to skeletal muscle in that they're both striated. They both have a sarcomere setup that leads to visual striations in the cells. But unlike skeletal muscle, which fuse together to form these large, what we call fibers, cardiac muscles retain their mononuclear morphology. Skeletal muscles are polynuclear. Cardiac muscles are mononuclear. And these intercalated discs have lots and lots of gap junctions, and that allows for cytoplasmic sharing. And so while the cells have not fused, they're still mononuclear, as opposed to the polynuclear skeletal muscles, they still overcome that technological barrier and achieve single unit contraction by using these gap junctions and these intercalated discs. Smooth muscle, like cardiac, is mononuclear, and they do also have a couple of ways that they solve that multi-unit to single unit problem. Again, they have to be able to contract as a single unit, and there are a couple of ways that smooth muscles do that. I'm not going to talk about that right now. I'll talk about it at the end of the chapter. But smooth muscle has a huge variety of types in the body. We see different organs with smooth muscle. These organs have different contraction patterns. Some organs stay contracted and then transiently release, so they can stay contracted for hours and days, whereas a skeletal muscle can barely stay contracted for 2 minutes. So we have different contraction patterns, we have different durations of contraction, and we have literally millions and millions of smooth muscles in our body. Every one of the goosebumps in your skin has a little band of smooth muscle called an erector pili muscle, and its contraction causes the goosebumps that you see on the surface of your skin. So a vast diversity of contraction patterns and locations are smooth muscle. Smooth muscle are generally not under direct conscious control, but that doesn't mean you can't initiate them with conscious intention. For instance, when you swallow, the muscles in your larynx and just above your epiglottis are skeletal muscle, and you initiate those voluntarily, But when you do that swallow, you initiate involuntary peristaltic wave leading to the stomach. So while it's not under direct conscious control, you can initiate smooth muscle contractions voluntarily. And that's why I stay away from voluntary and involuntary. I say skeletal, I say cardiac, I say smooth, because the voluntary and involuntary overlap, and the striation versus smooth also overlap. With skeletal muscle, we have some commonalities across all the couple hundred skeletal muscles that we have. They are usually attached to bones by tissue, connective tissue known as tendons. We have an orientation definition that many of you have seen in 305 and will again see in gross anatomy. That is to say, the more stationary bone that the muscle is attached to, or in some cases, the attachment point that is most proximal to the center line of the body is known collectively as the origin of the muscle. Though you do have things like the trapezius and the sternocleidomastoid where you have 3 attachment points. Again, the attachment point that's closest to the trunk or the most stationary bone is by definition the origin. Whereas the insertion is more distal from the center line of the body or the more mobile attachment. If you were to flex your bicep, what you see is that the top of the bicep, the one closest to your shoulder, doesn't move all that much, whereas the lower appendage of your arm does move. And so the origin would be up near your shoulder, and the insertion would be down by your elbow. We also can classify skeletal muscles by the type of motion that they achieve, the type of gross motion, like for instance bringing bones together. We call that a flexor. And so a bicep would be considered a flexor muscle, whereas the opposite muscle group on the other side of the arm, the triceps, are called extensors. They pull the lower arm away from the humerus bone. So the triceps would be considered extensors. That brings up a common theme in muscles, in that they work antagonistically. Muscles cannot push. Muscles can only pull. So in order to do pushing motions in the body, you have to not only have a contraction, you have a pulling motion across a lever and or what we call a fulcrum. A fulcrum is where you have a solid point and then you're moving a fixed lever across that point in order to achieve pushing motions as opposed to simply pulling motions. So these pushes and pulls that are different complex motions require not only the muscles contracting, the muscles can't push, remember. Muscles can only shorten and then relax. So we have to have these antagonistic muscle groups, but we also have to have fulcrums and levers. One more basic point about skeletal muscles is that they can regenerate if injured, depending on the severity of the injury. In relatively minor use-dependent type injuries, what we have are called precursor cells. These are called satellite cells. These are pre-muscle cells. They're partially differentiated, and in the face of cytokines from muscle tissue that has undergone repetitive contraction and or some minor injury, those satellite cells are then induced to divide and differentiate into new muscle fibers. And so here's just a picture of our flexor, our bicep. Again, here's the insertion down here. Here's the origin up here. We have our extensor group over here on the other side, the triceps. So flexion is moving bones closer together. In this case, the radius and the ulna being brought closer to the humerus. There's our origin, so forth. And then extension moves bones away from each other. So we're moving the radius and the ulna away from the humerus through contraction, again, across a fulcrum. So now we're going to start transitioning from the macro level of the muscle down into the cellular and then the subcellular levels of the muscle. At the macro level of the muscle, you've got connective tissue, you've got muscle fibers, you've got blood and nerves, you've got tendons, lots of things going on here, all wrapped up in this large thing that we call a muscle. And if you were to just grab your bicep right now, you would feel all of those bundles in there. And you can actually feel stripes in the muscle if you kind of squeeze down a little bit, you can begin to feel striping in there. Those stripes are actually known as fascicles. Those are bundles of muscle fiber that create that sort of linear feeling of ribbing, if you will, in your muscles. Fascicles contain connective tissue as well as individual large muscle fibers that are multinucleated as opposed to mononucleated like in the cardiac and smooth muscle. So an individual muscle fiber is a multinucleated cell. It's very, very large in the cellular world because it's a fusion of several individual cells. Well, when satellite cells come in, start developing new muscle fibers, they divide and then they fuse into this large Hindenburg, shall we say, sized cell called a muscle fiber. So a fascicle is a bundle of fibers, so it's the individual fibers plus some connective tissue. And then a muscle is a bundle of fascicles along with nerves and blood vessels and additional connective tissue and a little bit of fat as well. So the muscle is the macro. Fascicles are also macro. They're bundles of fibers. Okay, so fibers are individual muscle cells, if you will, that have been fused together. And so those cells are now one big giant cell. They've shared their cytosol at the most, shall we say, the most comprehensive way, whereas in smooth and cardiac muscle, they share cytosol, but in a different way than skeletal. So this slide right here is a great slide for terms, and you should know what each and every one of these terms mean or what these proteins and different structures do. So this is the language of chapter 12 to a certain extent. We certainly have extensor and flexor and those sorts of things, but a large percentage of the terms that you're going to need to know for chapter 12 are found right here on this slide. We certainly have the macro level, the skeletal muscle with connective tissue and fascicles, blood vessels, and nerves. Then we start to get ever so smaller. And when we get into an individual fascicle, we have the fibers, the cells themselves. And those fibers contain things like cytosol, and they contain specialized membrane structures that allow for contraction. We also have multiple nuclei because these cells have fused. So in that sarcoplasm, we have what's called sarcoplasmic reticulum. That's an endoplasmic reticulum. This is a calcium storage structure, calcium storage. We have the fibrils, if you will. These are the actin and myosin structures that give skeletal muscle and cardiac muscle its striated appearance. These are the effectors, if you will. And we have mitochondria, obviously to generate ATP, some other types of metabolism going on here. And then we have what are called inclusions. These are structures inside cells that don't have membranes. And the big inclusion in muscle is glycogen, which is a polymer of glucose. In those myofibrils, we have a whole bunch of different proteins. We have thin filaments, which are made up of troponin, actin, and tropomyosin. And we have thick filaments, which are made up of myosin primarily. And myosin is a very large protein that is involved intimately. It's a motor protein, so it's involved in contraction and extension. We have two other proteins that are called accessory proteins. They're called titin because of its large size, and nebulin, and we'll get to those functions a little bit later. All of these things are organized into a structure that we're going to learn in detail known as a sarcomere. So the sarcomere contains thin filaments and thick filaments, titin and nebulin. So these are the subcellular structures that we in particular need to know what's going on and what they're doing. And just so we make sure, the the terminology when it comes to muscles versus general cells came about because people were studying muscles and they thought it was so different in terms of specialization. They decided to call the cells fibers instead of just cells, even because they're multinucleate and they do have a very specialized structure. The cell membrane in a muscle cell is called sarcolemma. The sarcoplasm is the equivalent of cytoplasm, and then sarcoplasmic reticulum is the equivalent of endoplasmic reticulum. Similar functions as so forth. It was just that because of the level of specialization that these terms came about. We're just at the half-hour mark for this lecture, so I'm going to wrap this up for now. So this will end part 1A. We will finish off part 1 in part 1B coming up. Thanks for your time and good luck.